Welcome, everyone, to Genetics Essentials for Clinical Research Professionals, Part 2. In this webinar, we're going to be covering a little bit about the different genetic concepts that underpin precision medicine clinical trials and talk a little bit about how they relate to advances in precision medicine and how new technologies are also informing precision medicine clinical trials. But before I jump into our presentation, I'd like to spend a little bit of time talking to you about Sano, what we do, and introduce myself. Sano is a 360-degree platform for precision medicine study. Our main aim when we created the company was to accelerate the world's transition to precision medicine. One thing we quickly realized as academics, we were studying at the University of Cambridge at the Sanger Institute at the time, was that there was a lot of exciting research happening all around the world. The process by which we match patients into clinical trial, match patients ultimately with researchers so that they can be given access to treatments that are likely to work for us at the same time as increasing the odds of success of a clinical trial. Was just really complex and slow as a process. We created the Sano platform with the aim of facilitating this matchmaking. We cover the end-to-end process, starting with the strategic consultation, where we try to understand our clients' needs and success criteria for the trial in great depth, and where we assess the demographics of the patient population at stake, the biomarkers that have been selected for the trial, and recommend best practices in terms of how to set up the genetics and precision medicine part of the trial. We also provide patient finding and recruitment services, manage the biomarker testing, genetic sequencing, and other assays that could be relevant to the trial. We manage the long-term patient engagement by providing personalized content based on patients' genetic background or condition of interest, and generally speaking, by also providing them opportunities to engage in research and in clinical trials that are directly relevant to their conditions. And at last, throughout the process, of course, we analyze the data that's coming through, let it be in terms of patient recruitment and engagement, or in terms of the different biomarkers and data points that we're finding. A little bit more about me. I studied genetics and neuroscience at the University of Cambridge. My co-founder and I created Sano after we met during our PhDs. Personally speaking, I was focusing on the embryonic origin of childhood cancer, and I had the opportunity to look beyond traditional genetics technique, looking at gene expression and single cell technologies, which was really fascinating and opened my eyes to what the future of precision medicine could look like. Before doing my PhD at the University of Cambridge, I worked at JP Morgan in operations. And so as a result of my combined experience in science and in ops, today I lead business operations at Sano. And in particular, I also focus on the clinical and scientific operations. So anything related to genetic sequencing and to the different assays and biomarkers that we use to support precision medicine clinical trial. I'm also a mom of two little boys, and that is an important part of my journey because it's giving me even more motivation to build the foundations to transition in a world where we can match patients with the right treatment. Although rare diseases are rare, as the names indicate, 1 in 7 people in the world are affected. And so that's a very large number of individuals collectively. I have huge hope that the work that we're doing, and in particular the use of genetics and other assays into clinical trial, can dramatically improve our odds of finding treatments for these conditions. So today we'll be focusing on 3 core aspects. First off, I'll be talking a little bit about some genetics concepts that underpin diseases and conditions that we study. I'll be talking a little bit about the different types of genetic changes that we tend to observe and that tend to be associated with different conditions. I'll get into the genetics in clinical trial design and education part of this presentation, where we'll be talking about how these different genetic changes affect the potential modalities that we can explore in clinical trial. And of course, talk a little bit about why bringing in genetics and other biomarkers into clinical trial design tend to dramatically increase the success rate. And I'll spend a bit of time at the end as well talking about emerging technologies in the field of genetics and some innovations that we're seeing more and more into clinical trials, and talk about how this might be affecting the future of medicine. So I'll start diving down a little bit into genetic concepts in precision medicine. I think we're all aware at this point that there is genetic susceptibility to disease and that some people are more likely than others to develop a condition. In some cases, it's almost a direct mapping. If you have a specific genetic change, then it causes the condition itself. But in many, many cases, genetics is actually quite complex. And so what happens is that it can take one change or multiple change to increase the susceptibility of developing a disease, but is not often the case that it's 100%. Before I dive a little bit more into the gene environment interaction and other aspects of what increase the susceptibility to disease, I'd like to talk a bit more about the different type of genetic changes that we observe and how these are linked to specific condition. Today, I'd like to dive a little bit into the different genetic changes that actually underpin some of these conditions and explain a little bit the difference between them. Perhaps one of the simplest of the genetic changes that we observe is point mutation. Point mutation is simply a letter change in the genome. So usually from what we observe in the general population, it's a single nucleotide change, and it can lead to changes in the protein functions or no change at all. So when the change in the nucleotide doesn't lead to a change in protein function, then that's usually normal part of diversity in humans. However, when that single change starts affecting protein function and affects it negatively, it can result in specific conditions. So an example of this is sickle cell disease, which results from a single point mutation in the hemoglobin gene with all the symptoms that we know to be associated with. So again, point mutations happen fairly frequently in humans in general, and a normal part of diversity, but in the case where it affects genes where protein function is altered, can lead to some conditions. Insertions are slightly different. Instead of having just a change in the nucleotide in the DNA sequence, we have additions of one or more nucleotides into the DNA sequence. So we start having extra nucleotide in the genome. This can disrupt the reading frame, so it's frame shift mutation, which also, just like point mutations, can alter protein function. So an example of this are certain types of muscular dystrophy, which are caused by insertion in the dystrophin genes, for example. So as you can see, in both cases, the end result is that gene expression and protein function is disrupted as a result of the change, although the nature of the change itself is looking slightly different when you look at the genome. Deletions is kind of the opposite as insertion. Instead of having addition of nucleotide, there is a loss of one or more nucleotides from a DNA sequence. Again, just like insertions, this can cause frame shift mutations, and that can affect protein function, which leads to some conditions such as cystic fibrosis, for example. Deletion in the CFTR gene are associated with cystic fibrosis. Sometimes we see more complex chromosomal rearrangements as well, so it's not simple point mutation, insertions, or deletions, but we see large-scale structural changes in chromosomes. So that includes duplications, inversions, translocations, large chunks of deletions. This can often affect many genes at once and tends to have quite profound consequences on the phenotypic effects or the symptoms that we see in the patients. An example of that is the Philadelphia chromosome, which is a result of a translocation between chromosome 9 and 22, and which has been associated with chronic myeloid leukemia. So as you can see, all of these genetic changes, to some extent, affect the protein function of the cell. And when the disruption to the protein function of the cell is profound or is necessary to sustain life, we can start seeing symptoms or we can start seeing conditions arise. So why is there genetic variation really in humans? Why do the genetic changes that we presented above occur in people? Genetic changes underpin genetic diversity. I touched upon it a little bit when we talked about point mutation, but ultimately the human genome, is quite responsive and essential for populations to adapt to changing environments as a result, and so for the process of natural selection. Genetic changes is, what differs one human from the next, and also to a large extent, what has given humans the ability to adapt to their changing environment. But of course, some genetic changes can lead to undesirable effects and can lead to specific disease where protein function is disrupted and things don't function quite the way that we expect them to. This is where personalized medicine plays a big role. Understanding the genetic variation is super important to develop targeted therapies for those individuals that develop genetic changes that ultimately end up making them sick. Understanding exactly which gene is disrupted, which type of genetic change the gene is disrupted by, and exactly what gene expression process is disrupted as a result, what protein function is disrupted as a result, is fundamental for us to be able to identify therapies. I'll dive a little bit into gene regulation. I think, as you're all aware, the whole genome that makes an individual unique is the same throughout lifetime. So you have the exact same genes from the minute you are born until the last minutes of your life. However, what responds flexibly to the environment, the system by which genes actually respond to their environment, is gene regulation. Gene regulation is the process by which cells control the amount and the timing of gene expression, and so as a result, the amount and timing of protein expressed in the body. This allows organisms to respond to the environment, but also to differentiate into various cell types. You can imagine, for example, that a skin cell is pretty different from a heart cell. They all carry the same genome, so how do they manage to differentiate into the type of cell that they're due to become and hold the functions that they're due to hold? It's very much through gene regulation and gene expression. So I'll talk a little bit about the key elements of gene regulations. Promoters are DNA sequences that are located at the start of the gene. They act as docking sites for RNA polymerase and other proteins to initiate transcription. So this is the site at which transcription and gene expression starts. Enhancers are DNA elements that can be located far from the gene that they regulate, but they can increase the rate of transcription by ultimately enhancing the activity of the promoter. Silencers play an opposite role. DNA sequences that repress gene expression, ultimately, they can be located at various positions relative to the gene they control, and their role is to stop transcription. Transcription factors are proteins that bind to DNA sequences, so promoters, enhancers, silencers, to regulate transcription. They can either activate or repress gene expression. So as you can see, there are really clever ways for gene expression to be regulated, for transcription to start, to stop, to accelerate. Quite often, when we observe genetic changes that are linked to disease, these processes are altered, they're dysregulated. So sometimes transcription doesn't start at all when it should, or it's enhanced where it shouldn't, or on the other end, it can be silenced when it shouldn't. It's quite a complex machinery in any single cell. It's a tightly regulated process, and so disruptions to the genome can potentially also lead ultimately to disruptions in gene expression, gene regulation. So why do we have gene regulation processes in the human body? As mentioned earlier, it enables cells to differentiate and form complex organisms. So if we go back to our skin cell or heart cell example, what we will see is very different gene expression profiles between these two types of cells, despite having the same DNA inside the cell. It also supports adaptation and allows organisms to respond to environmental changes. So some genes might be upregulated or downregulated in response to environmental factors such as stress or pollution or others. But of course, dysregulation of these processes can also lead to diseases, including cancer and a multitude of other genetic disorders. Genetic linkage occurs when genes are located close to each other on the same chromosome, and so they tend to be inherited together more often than by chance. The closer two genes are, the less likely they are to be separated during crossing over and meiosis, making them genetically linked. So what are the principles then that underlie genetic mapping? The likelihood of two genes being separated during crossing over serves as a measure of the distance between them. It can be used to create genetic maps that represent the order and the relative distance between genes on the chromosome. And so these are all techniques that are used to understand genetic linkage and genetic changes, particularly useful to look at in the context of genetic conditions. Genetic markers are DNA sequences with known location on the chromosome, and they are used to track the inheritance of genes nearby. Again, they are useful in identifying genetic contribution to disease. So what are the applications of gene mapping? In the context of understanding specific disease, understanding genetic linkage and mapping the genes that are related to disease, researchers can actually identify predisposition and develop therapies. They may be able to understand that specific genetic changes are associated with higher odds of developing a disease. And when we understand which genetic changes are associated with a condition, we can start looking into the different gene pathways and the protein that they regulate. The hope behind this, of course, is that we can identify treatment potentially that increase the protein or reduce the protein that is affected by the downstream effects of these changes, or potentially go a little bit more upward in the treatment pathways and target the gene. We'll talk a little bit about that when we get into the different precision medicine modalities that we are currently exploring. Gene mapping is also interesting in evolutionary biology. Gene maps can help trace the evolutionary history of species by comparing genetic linkages. And of course, it's really important for personalized medicine. It enables the development of personalized treatment plans based on an individual's genetic makeup. And we'll talk about some examples of that in a minute. Now, genetics is complex, and we mentioned it earlier. Whilst there is sometimes a direct link between a change in the genome and a disease, more often than not, the genetic changes that we observe that are associated with disease are multifactorial. It means that there tend to be multiple genetic changes that combined together lead to the increased risk of condition rather than a single genetic change in the genome. And of course, that makes it a little bit more difficult for researchers to try and identify the cause. So in polygenic inheritance, multiple genes actually contribute to the final phenotype of a trait. So these genes may be located on different chromosomes entirely, and they interact in an additive or non-additive way. So some examples that we see in the day-to-day include height, skin color, eye color, where the contributions of many genes actually result in continuous range of possible phenotypes. But of course, polygenic inheritance is also something that we see a lot in chronic diseases that humans are affected by. If we think, for example, about liver disease, kidney disease, cardiovascular risks, more often than not, multiple genes and genetic changes combined together increase or decrease the risk of an individual. Another aspect of the complexity of genetic conditions is multifactorial disease, where gene and environment work together to cause an additive effect and increase the risk of developing a condition. So again, these are diseases that are influenced by multiple genetic factors, but also environmental interactions. These diseases do not follow a simple Mendelian pattern of inheritance. So for example, we were talking a little bit about it, but heart disease, diabetes, many types of cancer, whilst there may be genetic predisposition to develop these cancers, there also often needs to be some environmental triggers for them to happen in any individual. Many individuals who carry these risk factors will not end up developing the disease. And so we can do a little bit of refinement here when it comes to precision medicine to understand potentially individuals who are at higher risk and to understand what environmental factors are more likely to trigger the condition so that we can do some prevention. If you think about asthma, for example, again, it's a condition that is very much, that has some genetic factors associated to it. But at the end of the day, there also need to be some environmental triggers for asthma crisis to take place, for example, at least in some forms of asthma. I'll get to the next slide. So I'll touch a little bit upon the key concept in these more complex patterns of inheritance. We discussed it a little bit in the previous slides, but ultimately there is the genetic susceptibility, which is some changes in the genes, let it be point mutation, insertion, or deletions, as discussed above, that increase the risk of developing multifactorial disease, and often, particularly in the context of polygenic risk, it's multiple genes together that increase that risk. But they do not guarantee that the individual will develop the disease. There is then an environmental influence, for example, with external factor that can significantly impact then the likelihood of the disease developing, particularly so in these individuals that have increased genetic susceptibility. So here we're thinking about, for example, pollution, stress, diet, exercise, and so on. And so together, the genetic susceptibility and the environmental influence can help us understand risk better for a single individual and put in place personalized prevention strategies. Now, why does it matter? From a personalized medicine point of view, tailoring prevention and treatment strategies based on an individual's unique genetic makeup and environmental exposure is much more likely to result in successful intervention. It's also much more likely to result in adherence, because if someone understands that the risk applies to them in a specific way, more so than to the general population, they're also more likely to take on board particularly lifestyle changes advice. But in some cases, there may also be medications or treatments that can be taken to slow down symptom progression or to prevent a condition altogether. And of course, it matters for public health. Identifying at-risk population means that we can develop strategies to mitigate the impact of multifactorial disease and potentially get these individuals diagnosed a lot sooner. So let's talk a little bit about the gene-environment interactions. Gene-environment interactions describe the complex way in which genetic and environmental factors collaborate to influence traits and disease. And these interactions underscore just how difficult it is to attribute specific traits or disease to genetics alone. But that's not to say that it doesn't play an important role. There are several mechanisms of interaction. Environmental factors can activate or suppress gene and alter their expression and potentially lead to disease. So for example, we know that there are different mutations in serotonin receptors that lead people to be differently susceptible to stress in the way they respond to stress and in the damages that it may cause on their body. There is also an element of sensitivity. Individuals with certain genetic profiles may be more sensitive to environmental influences, such as pollutants, disease, such as pollutants, diet, or stress. Multiple gene mutations can interact in a complex way, affecting disease severity or presence and highlighting the complexity of genetic factors in disease. This is genetic epistasis. A single gene can influence multiple disease as well, as seen in multiple conditions like diabetes, and that shows the interconnectedness of the genetic contributions to health. So again, a disrupted gene sometimes regulates multiple functions in the body and is going to be leading to a range of symptoms that doesn't actually cover a single disease category, but might make the individual more susceptible to multiple conditions. Many diseases are caused by gene and gene-environment interactions. We talked a little bit about asthma before, but it's also the case for obesity. There are some genetic predispositions that interact with lifestyle factors such as diet and physical activity. Both of them together influence the risk of obesity. So that leads us to really the interesting part of this. What can we do with this information? How can this information really be transformational in the way we treat patients and in the way we lead clinical trial? Pharmacogenomics combines genetics and pharmacology to understand how an individual's genetic makeup affects their response to drugs. There are two ways in which this can be really interesting. The first, of course, is by increasing the efficacy of this treatment. If you take a clinical trial where only a subset of patients have a mutation, let's take an example. Let's look at ALS, for example. Several people develop ALS on a given year. Out of those people, only a subset will have a genetic mutation that we know today is associated with the risk of developing ALS. The others might have other genetic mutation that we haven't yet discovered, but ultimately within these genetic mutations, some pharma companies are trying to develop treatments that affect a specific genetic mutation within the subset of patients. I'm going to talk about SOD1 here. SOD1 is a gene that has been associated with the risk of developing ALS. And so some pharmaceutical company and biotech company out there are currently developing treatments that target the SOD1 gene. Now, as you can imagine, whilst this treatment might be successful in patients that have genetic changes in the SOD1 gene, they might not be successful in patients with ALS who do not have this, these changes in SOD1 or have changes in other genes that are related to ALS. And that's interesting because traditionally we might have given a treatment to a group of people with ALS or with a specific condition and find that overall the treatment didn't work quite well, although a small group of patients would have seen positive results with treatment. This is genetic variability, and this is what pharmacogenomics enable us to do. By looking specifically at the individual's genetic makeup, it can enable pharmaceutical company biotechs, researchers to identify molecules that can potentially target the pathway in individuals with a specific mutation. And conversely, developing a potential molecular target can work really well in these subgroups, but might not work well in others. By being able to understand the genetic makeup of the patients that are getting into clinical trials, we are able to identify people who are most likely to actually benefit from the treatment they are being given. And again, as a result, this increases the odds of a potential treatment passing the bar for significance and being made available to others with similar genetic mutations into a wider public. The other really exciting side of pharmacogenomics is the ability to predict side effects. When treating a patient, there always is a cost-benefit analysis to be made. Ultimately, a treatment should be prescribed if the benefits outweigh the risks. And so, again, with the former, we calculate the odds that the treatment has of being successful. With the latter, we calculate the odds that a patient is likely to develop side effects. We know, for example, that different people metabolize drugs in different ways. There are several very successful examples of that in the literature. We know that a subset of individuals, for example, respond quite dramatically to warfarin, which is a blood thinner, and can develop internal bleeding, for example, as a side effect. It's a very rare side effect, but it's one that you really want to be taking into account when you're prescribing this sort of medicine, and being able to identify individuals that are most likely to develop these side effects is important, because we can either decide that the treatment is not right for them, or simply get similar results as in other people by reducing the dose significantly to minimize side effects. So that leads me to genetics in clinical trial design and execution. How do we take all of this background in terms of the different aspects of genetics that influence disease risk and use the techniques that we have available to increase the odds of clinical trials being successful and to increase the odds of treatment being rolled out to patients who currently do not have access to successful treatment for their conditions? As mentioned earlier, a big role of genetics and precision medicine in clinical trial is to identify and utilize genetic biomarker to actually ensure that the treatment that is being tested is being given to people who are more likely to respond to it and who are the least likely to develop side effects as a result of it. So this is how it works. Having genetic sequencing and other assays, depending on the context of the disease as well, can help with targeted patient selection. Identifying participants with specific biomarker increases the likelihood of demonstrating treatment effectiveness. Another way that we can do that is through dose optimization. Some clinical trials fail because there is quite a lot of variation between individuals in the dose of the medicine that they need to respond to treatment. Generally speaking, the right dose is the minimum possible dose that someone should take to be seeing the full effect of the treatment in terms of reduction of symptoms without increasing the odds of side effects. And this dosage is quite different from one person to another. So genetics enable us to forecast a little bit better which groups of patients are more likely to need higher dosage or lower dosage in a much better way than, just height and weight can. And at last, biomarkers can be used to monitor treatment response in real time. So we've talked a little bit about how when we have a drug target that is specifically designed for a genetic change, it increases the odds of success. But I do genuinely believe that having genetic information supporting clinical trial is useful even in those trials who haven't yet identified a precision medicine angle to the drug they are testing. Having the genetic data means that we can look back into the data and see whether patients who have developed side effects or patients who have not responded or responded particularly well to the treatment have some genetic, commonalities, if you will, between them, and whether that can help us understand a little bit better the populations in which this is likely to be successful. I'm going to give a few examples of some key trial in precision medicine that used some of this principle and explain how they were successful in using this data to advance medicine. The first one I'd like to talk about is TAILOR-X. TAILOR-X was a landmark study that focused on women with axillary lymph node negative breast cancer. In this group of patients, obviously, doctors tend to be quite conservative, and to increase the odds of survival tend to prescribe both hormonal treatment and chemotherapy, with the idea that, the sort of more you throw at it and the less likely this woman is to have a cancer recurrence. But of course, we know chemotherapy comes with a huge amount of side effects and affect the quality of life of these patients. And so here the study was trying to stratify patients based on the 21 gene expression test to decide which women were most likely to get cancer recurrence versus the one that couldn't, weren't likely to develop a recurrence. So the trial was pretty significant because it identified subgroups of patients based on gene expression that could actually safely avoid chemotherapy without an increased risk of recurrence. Another trial I'd like to speak about is the I-SPY2 trial. Here again, it was an ongoing study for patients with newly diagnosed locally advanced breast cancer. And this is a really, really good example of adaptive design for clinical trial. The way that the trial was set up meant that the study used biomarkers to identify which therapy were likely to be the most effective for specific patient subgroups, and that was based on their tumor characteristics. That allowed for multiple investigational drugs to be tested at the same time within the same trial, with the most effective drugs graduating then to later phase trials based on how effective they were in specific biomarker-defined subgroup, and ultimately advancing our understanding of medicine by bringing in that stratification. Here we were looking at the tumor characteristic. Cancer is obviously complex because we often have to take into account the genetic makeup of the individual and the genetic makeup of the tumor. But in this case, the sort of precision medicine angle of this trial enabled multiple therapies to be tested at once, so that only the best ones could ultimately be brought into later phases of clinical trial. Now, it's particularly important when leading a precision medicine clinical trial to have strong safeguarding privacy and setups in place and to empower participants. Genetic-based clinical research is a fast-evolving field, and there have been many ethical considerations around genetic data privacy and informed consent that have emerged over the year, and for all the right reasons. When it comes to genetic data privacy, participants who enroll in clinical trials that involve genetic sequencing need to have the right amount of support and to have the confidence that their genetic data is protected by very strong protocol, that there are strong genetic data privacy agreements in place. And of course, they need to feel comforted that this won't affect their future chances of accessing treatment or health insurance in some countries. Likewise, it's really important to have informed consent. In the early days of genetic clinical trial, I think the experience of patients being sequenced for the first time often was that maybe they hadn't been quite as prepared as they wish for what they were going to learn and the impact it would have on their family. In some cases, of course, they only consented to their gene being sequenced for one purpose and their data was used for other purposes. And likewise, in another extreme example of that, some programs consent patients only for one study and don't really have the infrastructure or the software in place to then recontact them and to be able to offer them the opportunity to contribute to research on a more wider scale. And as a result, a lot of the initial work that has been done in sequencing these patients and in obtaining some responses from them on their history, etc., is lost because it can't be reused for a wider cause. So at Sano, we obviously take both very seriously. We have a dynamic consent model in place where ultimately we make sure that the consent model is crystal clear for participants and that they understand exactly who is going to see their data, who is going to access it and in which context. But we also have the software and infrastructure to be able to recontact participants if a relevant study or clinical trial comes down the road a few months later. We are able to once again acquire consent for what we want to be looking at here, with the patient being put at the center of this decision making. Again, informed consent requires the right amount of information. And so in the case of a pretty complex disease or conditions that really affect the quality of life or may affect family planning decisions, we also support patients with pre and post genetic counseling to ensure that they are supported in understanding exactly what the clinical trial means for them and what they may learn from their data. I'd like to end this presentation by going a little bit into emerging technologies and innovations in genetics, because this is probably one of the most exciting area of medical research today. And I'll talk a little bit about CRISPR-Cas9. CRISPR-Cas9 is a technology that has emerged as a groundbreaking tool, offering unprecedented precision in editing DNA sequences. So effectively, we're talking about gene editing here in humans. This innovation is driving the development of therapies where we are discovering treatments for conditions that we previously thought were completely incurable. To give you a little bit of an oversight on how that works, ultimately, if you have a genetic change in the gene that then leads to downstream dysfunction in gene regulation and ultimately in protein function, what pharmacogenetics typically tries to do is to target the protein or some upstream elements of the gene regulation process pathway, so that ultimately the protein function can be restored to the best possible extent. And of course, this is a very important way of treating disease. But gene editing goes all the way back to the genome. And so if we take, for example, our point mutation example that we covered at the start of the presentation, with some work on precision, etc., what gene editing can do is ultimately change back this nucleotide to what we see in a general healthy population. In the case of deletion, of course, it can replace the missing nucleotides. And in the case of insertion, in theory, it can remove the additional nucleotides that have made their way into the genome. That's, of course, hugely exciting because it's a far more permanent solution than constantly trying to target a dysfunctional protein function. Here, we're fixing the problem at the root cause in theory. It's obviously an emerging technology. There is still a lot of work that is being done on the accuracy of the technology and trying to understand whether, potential side effects would be introduced as a result of it. But there have already been successful uses in treating conditions like sickle cell disease and beta thalassemia by correcting the faulty genes responsible for this disease. It's easier at the moment to use CRISPR-Cas9 in genetic changes such as point mutation, more so than in very large genetic changes. But it's hugely exciting to see some applications for it already. There is a lot of hope as well that CRISPR-Cas9 is going to help develop personalized cancer treatment, for example, by editing immune cells so that they can target and destroy the cancer cell in a specific individual more effectively. There are some use cases as well with infectious disease with potential applications in disabling the genes of viruses like HIV, which express in human cells, and therefore offering new pathways for antiviral therapies. So again, we just really at the cusp of starting to use this technology in the context of human health, but it is opening the door for precision medicine trial that are going to be looking at using CRISPR-Cas9 technology to ultimately go back to the root cause of what led to the phenotype to start with, and instead of targeting downstream effects to fix the problem at the root cause. It's also, of course, a little bit more complex in genetic conditions that are polygenic in nature, and even more so in the ones that have huge gene environmental applications, as we discussed earlier. The more straightforward use for CRISPR is when there is a direct link between the genetic change and the symptoms and the condition and the phenotype. But of course, the extent to which we can apply it remains to be seen, and it's a hugely exciting field. Another emerging area, of course, in precision medicine is transforming discovery and treatment through the use of AI. AI is at the forefront of precision medicine at the moment. It excels at data analysis. It seems to be able to process and interpret vast amounts of data generated by genetic sequencing, and sometimes being able to identify patterns that even some of the best scientists might have missed, and to potentially understand a little bit better the linkage as well between different genes and pathways and how they relate to one another. I have huge hope that AI is going to help identify more patterns, particularly in multifactorial and polygenic disease, potentially linking the function of these genes in a way that we hadn't previously anticipated. AI is also very good for developing predictive model and certainly getting better at it for genetic disease, ultimately identifying who might be at a bigger risk of developing a condition, and identifying genetic changes here that might be at the root cause of it, so that we can enable early diagnosis. And at least, of course, the hope is that in due time, with AI becoming more performant, it might be able to come up with personalized treatment plans. There are multiple applications to this. One of the ways that AI could potentially contribute to that last part is by identifying drug targets, but also potentially in just combing through existing drugs on the market and trying to understand how they could be reapplied to different conditions and genetic changes by looking at the genetic data of individuals that have responded well to these drugs, or that may have shared pathways that could be targeted by existing drugs. So what are the current applications and the future potential? As we discussed, gene therapy research enables us to accelerate the discovery of targets through rapid screening of genetic mutation and to understand their impacts on disease. Pharmacogenomics are enhancing drug development plan and therapy selection by predicting individual responses to medications based on an individual's genetic makeup, which is much more accurate than other demographic data. And at last, all of these tools can help us assess the genetic risk factors for disease and offer new opportunities for prevention and early intervention. So as you can see, the different genetic changes that underpin disease are complex on their own, but when you add on top of it the very complex way in which genes regulate gene expression, downstream protein pathways, how they also interact with one another and how they respond to their environment, and taking all of these different data points in trying to understand diseases is complex, but bringing in these data points into clinical trials is a hugely exciting step in the transition to precision medicine, because it enables us to have better visibility on who is more likely to respond well to a new treatment, who is more likely to develop side effects. And with technologies like CRISPR-Cas9 and AI coming into play, we at Sano have a huge hope that precision medicine might be able to unlock treatments for conditions that we thought were previously incurable, and accelerate dramatically the rate at which clinical trial can successfully identify drug targets or other treatments that might be able to help find a cure for these conditions. So thank you very much for listening to this webinar, and I'm happy to take any questions.